Top 50 Pharma interview questions on liquid dosage forms, solutions, suspensions, and injection.
General concepts.
What is the difference between a solution and a suspension? A solution is a homogeneous mixture where the solute is completely dissolved in the solvent. In contrast, a suspension is a heterogeneous mixture in which particles are dispersed but not dissolved. Solutions appear clear, while suspensions may be cloudy. Suspensions require shaking to ensure even distribution before use. Both forms are essential in pharma for delivering drugs effectively. What factors influence the stability of liquid dosage forms? Stability is influenced by temperature, pH, light exposure, and the type of container. Ingredients like preservatives help prevent microbial growth. Antioxidants are added to reduce oxidation. In suspensions, particle size and viscosity impact stability. Each factor ensures the drug maintains effectiveness over its shelf life. Explain the importance of pH in liquid formulations. pH affects the solubility and stability of drugs in solutions and suspensions. An optimal pH prevents degradation and maintains the drug's potency. For certain drugs, pH ensures they are non-irritating when administered. pH adjustments also improve drug absorption. Buffer systems are commonly used to maintain pH. How do solutions differ from emulsions and suspensions? Solutions are clear mixtures with dissolved particles, while emulsions contain two immiscible liquids, like oil and water. Suspensions have undissolved particles that require shaking. Emulsions and suspensions are typically cloudy, whereas solutions are clear. Each form is chosen based on the drug's properties and the required delivery method. What is solubility, and why is it important for solutions? Solubility is the ability of a substance to dissolve in a solvent. High solubility ensures that a drug is evenly distributed in a solution. This is crucial for accurate dosing and patient safety. Poorly soluble drugs may require special formulation techniques. Improving solubility often involves co-solvents or pH adjustments. How does particle size affect the stability of suspensions? Smaller particles in suspensions are less likely to settle quickly, which improves stability. Larger particles tend to sediment faster, leading to non-uniformity. A consistent particle size ensures each dose has the correct amount of medication. Particle size also affects the drug's absorption in the body. Milling and homogenization are used to achieve the desired particle size. What are co-solvents, and why are they used in liquid formulations? Co-solvents enhance the solubility of drugs that poorly dissolve in water. Common co-solvents include alcohol and propylene glycol. They make drugs stable and uniform in solutions. Co-solvents are chosen based on the drug's properties and intended use. They help achieve the right drug concentration for effectiveness. Explain osmolarity and its importance in injectable solutions. Osmolarity measures the concentration of solutes in a solution. For injectables, osmolarity should match the body's fluids to avoid irritation. Hyperosmolar solutions can cause cell damage, while hypoosmolar solutions may lead to swelling. Maintaining proper osmolarity ensures patient comfort and drug efficacy. This is especially critical in fore and ophthalmic solutions. Why is viscosity important in suspensions? Viscosity helps suspend particles in a liquid, reducing sedimentation. Higher viscosity means particles settle more slowly, creating a stable suspension. However, excessive viscosity can make a suspension hard to pour or inject. Viscosity also affects how the body absorbs the drug. Polymers and gums are commonly used to adjust viscosity. Describe the common types of solvents used in liquid formulations. Water is the most common solvent, often purified for quality. Alcohol is used for drugs needing higher solubility. Oils like sesame oil are used in injectables for slow release. Co-solvents like propylene glycol enhance stability and solubility. Solvent choice depends on drug properties and formulation needs. Formulation and ingredients. How would you choose a preservative for a liquid formulation? The choice depends on the formulation's pH, compatibility, and intended use. Common preservatives include parabens and benzyl alcohol. They prevent microbial growth in multi-dose containers. Preservatives are essential for patient safety in liquid formulations. They must be safe and non-irritating for their application. What are surfactants and what role do they play in suspensions? Surfactants reduce surface tension, helping particles disperse evenly. They prevent particles from clumping together, which stabilizes the suspension. Common surfactants include polysorbates and lecithin. Surfactants are essential for maintaining uniformity in suspensions. They also improve drug absorption in certain formulations. Explain the use of buffer systems in liquid formulations. Buffers maintain a stable pH, which helps preserve the drug's stability. They prevent pH changes that could degrade the drug or cause irritation. Common buffers include phosphate and citrate systems. Buffers are particularly important in injectables and ophthalmic solutions. Maintaining the correct pH ensures drug safety and effectiveness. What role do sweeteners and flavoring agents play in oral solutions? They improve the taste, making the medication more acceptable for patients. Sweeteners like sucrose and artificial sweeteners mask unpleasant flavors. Flavoring agents are added to cover medicinal tastes, especially in children's formulations. A pleasant taste can improve patient compliance. Both sweeteners and flavors are chosen based on safety and stability. How can you improve the bioavailability of a poorly soluble drug? Techniques include using co-solvents, reducing particle size, and forming salt forms. Emulsions or nanoparticles can improve solubility and absorption. Surfactants enhance wetting, making drugs more bioavailable. Solid dispersions are also used to improve solubility. Bioavailability ensures the drug reaches therapeutic levels in the body. Describe the use of suspending agents in pharmaceutical suspensions. Suspending agents increase the viscosity of the liquid, helping to keep particles evenly dispersed. They prevent sedimentation, so the suspension remains stable over time. Common agents include gums like xanthan and cellulose derivatives. The choice depends on compatibility with other ingredients and the drug's requirements. Proper suspending agents ensure accurate dosing and product stability. Why is sterility crucial for parenteral solutions? Parenteral solutions are injected directly into the body, bypassing the skin barrier. Any contamination can lead to serious infections or adverse reactions. Sterility ensures the solution is safe for injection and free from harmful microbes. Rigorous sterilization techniques like autoclaving and filtration are used. Maintaining sterility is essential for patient safety. Explain the process of adjusting tonicity in solutions. Tonicity refers to the concentration of solutes relative to body fluids. In solutions, tonicity is adjusted to prevent irritation when administered, especially in injectables and eye drops. Isotonic solutions match body fluids, minimizing discomfort. Sodium chloride or other agents are added to achieve isotonicity. Proper tonicity ensures the solution is comfortable and safe for use. What are humectants, and why might they be used in topical solutions? Humectants are ingredients that attract moisture to the skin. They keep topical solutions from drying out and improve the product's stability. Common humectants include glycerin and propylene glycol. In skincare or wound care solutions, humectants improve skin hydration and comfort. They also help enhance the absorption of the active ingredient. Describe the role of antioxidants in liquid dosage forms. Antioxidants prevent oxidation, which can degrade the active ingredient over time. They help maintain the stability and shelf life of the product. Common antioxidants include ascorbic acid and tocopherol, vitamin E. In solutions, antioxidants are essential for preserving potency. They protect the drug from reacting with oxygen and losing effectiveness. Manufacturing processes. What equipment is commonly used for mixing and homogenizing suspensions? Mixing and homogenizing equipment ensures particles are uniformly dispersed. High-shear mixers, colloid mills, and ultrasonic homogenizers are common tools. This process prevents particle clumping and ensures a stable suspension. Proper mixing is essential for product quality and uniform dosing. Different equipment is used depending on the formulation requirements. Describe the sterilization process for injectable solutions. Sterilization removes or kills all microorganisms in injectable solutions. Techniques include autoclaving, filtration, and irradiation. Filtration is often used for heat-sensitive drugs as it removes microbes without heat. Each method is chosen based on the drug's properties and formulation stability. Sterilization ensures the solution is safe for injection. How do you minimize foaming in the manufacturing of liquid formulations? Foaming can trap air, affecting product quality and consistency. Techniques to minimize foaming include adjusting mixing speed and using anti-foaming agents. Vacuum processing also helps remove air from the mixture. Excessive foam can lead to inaccurate dosing and stability issues. Controlling foaming is essential for maintaining quality. What is the difference between hot and cold processing for solutions? Hot processing involves heating ingredients to dissolve or disperse them, useful for viscous materials. Cold processing is done at room temperature, preserving heat-sensitive ingredients. Both methods are used based on the formulation's needs and ingredient properties. Hot processing may improve solubility, while cold processing prevents degradation. Choosing the right method ensures the formulation's stability and effectiveness. What are the critical parameters for the lyophilization of solutions? Lyophilization, or freeze-drying, is used to make stable, dry forms of solutions. Critical parameters include freezing rate, vacuum level, and drying temperature. These parameters ensure moisture is removed without degrading the product. Lyophilized products are reconstituted before use, often in injectables. Proper control of these parameters ensures product stability and efficacy. Describe the process of filtration in sterile solution manufacturing. Filtration removes particles and microbes from solutions, making them sterile. Sterile filters, typically with a pore size of 0.22 microns, are used for this process. This is essential for solutions that cannot withstand heat sterilization. Filtration ensures the solution is safe for administration. The process must be validated to confirm effectiveness. How is air entrapment in suspensions handled during manufacturing? Air entrapment can lead to stability issues and impact product quality. Deaeration techniques, such as vacuum application or slow mixing, are used to minimize air bubbles. Anti-foaming agents can also help reduce trapped air. Entrapped air can cause sedimentation or instability in suspensions. Proper handling ensures a stable and effective product. Explain the significance of mixing time and speed in liquid formulations. Mixing time and speed affect how well ingredients are blended and dissolved. Too high a speed can introduce air, causing foaming, while too slow may lead to incomplete mixing. Proper time and speed ensure uniformity and stability in the formulation. They also impact particle distribution in suspensions. Accurate mixing helps maintain consistent product quality. What is granulation, and is it relevant to suspensions? Granulation is the process of forming particles into larger granules, primarily for solid dosage forms. In suspensions, it's less relevant, as particles are dispersed in liquid. However, granulation is important in pre-formulation when making solid ingredients for suspensions. Proper particle size is achieved through other means, like milling. Granulation ensures uniformity in solid formulations. What are critical quality attributes, CQAs, in liquid manufacturing? CQAs include aspects like pH, particle size, and sterility that impact product quality. They are identified based on regulatory guidelines and product requirements. Controlling CQAs ensures the formulation meets safety, efficacy, and stability standards. Examples include viscosity, homogeneity, and preservative effectiveness. Monitoring CQAs ensures consistent, high-quality products. Quality control and stability. How would you perform stability testing on a suspension? Stability testing involves storing the product under various conditions to monitor changes over time. Tests include measuring pH, viscosity, and particle size, as well as observing physical changes. Samples are checked at intervals to ensure the product remains stable. Environmental factors like temperature and humidity are varied. Stability testing confirms shelf life and storage conditions. What tests are necessary to ensure the homogeneity of a suspension? Homogeneity testing checks that particles are evenly dispersed in the suspension. It involves sampling different parts of the batch to check particle distribution. Microscopy and sedimentation rate tests are often used. Ensuring homogeneity is vital for accurate dosing. Testing confirms the product's uniformity across doses. Explain the importance of particle size analysis in suspension QC. Particle size affects stability, homogeneity, and bioavailability. Smaller particles settle more slowly, creating a more stable suspension. Particle size analysis uses techniques like laser diffraction to measure consistency. Ensuring the right particle size improves drug absorption and uniform dosing. It's a critical quality check for suspensions. Describe the procedure for microbial testing of solutions. Microbial testing ensures the solution is free from harmful microbes. Samples are incubated on growth media, and any microbial growth is measured. Tests like sterility testing and microbial limit testing are commonly used. Preventing contamination is essential, especially in injectables and ophthalmic solutions. Regular microbial testing ensures safety for patient use. How is sedimentation rate measured and controlled in suspensions? Sedimentation rate indicates how quickly particles settle in a suspension. It's measured using visual observation or analytical instruments. Suspending agents can slow sedimentation, making the suspension more stable. A controlled sedimentation rate ensures uniform dosing with every use. Stability testing helps determine the optimal formulation. What are the typical tests for checking the pH of a solution? pH is typically measured using a calibrated pH meter, which gives an accurate reading of the solution's acidity or alkalinity. For stable results, the pH should be within a specified range to ensure drug stability. pH indicators can be used as a quick visual check, although they are less precise. Regular pH checks are important, especially during stability testing. The pH must remain constant to prevent degradation and maintain the drug's effectiveness. Explain the process for measuring viscosity in liquid formulations. Viscosity is measured using a viscometer or rheometer, which assesses the liquid's resistance to flow. This property affects the stability and pourability of suspensions and solutions. Different types of viscometers are chosen depending on the liquid's thickness, such as capillary or rotational viscometers. Viscosity control is crucial for suspensions to prevent rapid sedimentation. Consistent viscosity ensures uniform dosing and ease of administration. How do you test for the presence of preservatives in solutions? Testing for preservatives involves analytical methods like high-performance liquid chromatography, HPLC. This test confirms that the preservative concentration is within the specified range, ensuring microbial protection. Regular testing during shelf life is done to check for degradation. Preservatives are necessary to prevent contamination, especially in multi-dose products. Accurate preservative levels are crucial for product safety. What is the impact of temperature on the stability of liquid formulations? High temperatures can speed up degradation, while low temperatures may cause precipitation or separation. Stability testing under different temperature conditions, accelerated and real-time, helps predict a product's shelf life. Temperature-sensitive formulations are stored in controlled environments, such as refrigerators. Temperature control is especially critical for biologics and injectables. Understanding thermal stability helps set safe storage guidelines. How is oxidation prevented in solutions? Oxidation can degrade active ingredients, causing loss of potency. Antioxidants like ascorbic acid or tocopherol are added to solutions to prevent oxidation. Proper storage in airtight, opaque containers also reduces oxygen and light exposure. Deaeration techniques are sometimes used to remove oxygen during manufacturing. Preventing oxidation is essential for maintaining product effectiveness over time. Regulatory and compliance. Describe the FDA guidelines on the manufacture of liquid pharmaceuticals. FDA guidelines cover good manufacturing practices, GMP, ensuring product quality and safety. They outline requirements for facility cleanliness, equipment, personnel training, and documentation. Liquid pharmaceuticals must meet standards for purity, potency, and sterility. The guidelines also mandate stability testing and record-keeping for quality assurance. Following FDA guidelines ensures compliance and product approval. What are good manufacturing practices, GMP, for liquid formulations? GMP guidelines ensure consistency, quality, and safety in pharmaceutical production. For liquids, GMP covers sanitation, equipment maintenance, and environmental control. GMP also involves batch records, personnel training, and standard operating procedures, SOPs. The goal is to minimize contamination and ensure product integrity. Adherence to GMP is mandatory to produce safe and effective products. How do you validate the sterilization process for injectables? Validation involves testing to confirm that the sterilization process consistently removes contaminants. This includes verifying temperature, pressure, and time parameters in autoclaving or filter integrity in filtration. Biological indicators like spore strips can test sterilization efficacy. Regular validation ensures the process meets regulatory standards. Validating sterilization is essential for patient safety. What documentation is required for liquid formulation approvals? Documentation includes formulation data, stability studies, manufacturing protocols, and quality control results. Regulatory bodies require proof of safety, efficacy, and compliance with guidelines. Detailed records of ingredient sources, batch records, and analytical data are essential. Validation reports and SOPs for each manufacturing step must be included. Proper documentation supports product approval and traceability. What is the ICH guideline on stability testing for solutions? ICH guidelines specify stability testing conditions, such as temperature and humidity ranges, to predict product shelf life. For solutions, stability tests must monitor changes in pH, color, potency, and microbial growth over time. Both accelerated and long-term studies are conducted. Stability testing ensures products meet quality standards throughout their shelf life. ICH guidelines are internationally recognized for regulatory submissions. Explain labeling requirements for solutions and suspensions. Labels must include the drug name, concentration, dosage instructions, and storage conditions. They also indicate the expiration date, lot number, and manufacturer details. Warnings and precautions are required for certain drugs. Labels help ensure the correct use and storage of the medication. Accurate labeling is essential for regulatory compliance and patient safety. How do you manage batch-to-batch variability in suspensions? Consistent raw material quality and controlled manufacturing processes reduce variability. Key factors include precise particle size, mixing time, and equipment calibration. Quality control tests confirm uniformity between batches. Batch records document every production step to trace sources of variability. Maintaining consistency ensures patients receive the correct dosage in every batch. What regulations govern the storage and handling of liquid pharmaceuticals? Storage guidelines vary by product type, including temperature and humidity controls to prevent degradation. Regulations require that facilities store products under controlled conditions with regular monitoring. Temperature-sensitive products may require cold chain storage. Proper handling minimizes contamination risks during transportation and storage. Compliance with storage regulations ensures product efficacy and safety. What stability data is required for a new liquid product submission? Stability data includes results from accelerated and long-term testing at specified conditions. Tests measure physical changes, potency, pH, and microbial growth over time. Data supports the proposed shelf life and storage conditions. Stability studies ensure the product remains effective and safe for its duration. Regulatory agencies require this data for approval. Explain the role of the European Pharmacopoeia in liquid formulation standards. The European Pharmacopoeia provides quality standards for drug formulations, including liquids. It defines specifications for ingredients, testing methods, and acceptable limits. Following these standards ensures consistency, safety, and compliance in European markets. For solutions and suspensions, the standards cover purity, pH, and microbiological limits. Compliance with the Pharmacopoeia ensures product quality across EU countries. Thank you for watching our video on the essential interview questions for liquid formulations, including solutions and suspensions. We hope this content has been valuable, providing you with insights and boosting your confidence for your next pharmaceutical interview. Whether you're preparing for a career in formulation, quality control, or regulatory affairs, understanding these foundational topics is crucial for success in the pharmaceutical industry. If you found this video helpful, please hit the like button and share it with others who might benefit from this knowledge. And don't forget to subscribe to PharmaTalks. By subscribing, you'll stay updated on the latest in pharmaceutical sciences, including formulation tips, regulatory updates, and career insights tailored for pharma professionals and students alike. We're committed to delivering high-quality content that supports your journey in the pharma field. so turn on notifications to ensure you never miss an update. Thank you for being a part of our growing community at PharmaTalks. We look forward to supporting your growth and success in the industry.